Good afternoon, everyone. It's a real pleasure to have you all here back at the liver seminars today. And it is a great pleasure today to introduce you to a good friend and a great scientist, as you'll hear now during the session, Fotis. And before I start and present him, I would just want to clarify a few technical points, and this is regarding the Q&A session. If you have any questions that start to mount during the presentation, please write them down on the Q&A. Or if at the end of the presentation you want to ask them personally and voicely, so just raise your hand, and the technician will give you permission to speak, and then you can ask them live. Okay, so that's how it's going to work. Now, I want to thank Fotis for being here today. Again, it's a great pleasure. Thank you for accepting to be here and giving your presentation. I know you're always short on time between patients and the other type of patients in the lab. So it's always a rush. But before I give the word to you, so I just want to present you. And so Fotis, for who doesn't know him, he's an MD, which was trained in Greece at the National and Kasplotidrian University of Athens. Then he went to the UK, and where he kept going with his training in gastroenterology. He got also certificate in medical education in 2012. He got his PhD in surgery from the University of Cambridge in 2017. And he completed specialty training in gastroenterology and hepatology in 2020. He's currently a group leader in the Cambridge Stem Cell Institute and an honorary consultant in hepatology at the Cambridge University Hospital. So this is, I think, a best case scenario where you really try to do the scientific part, the research, but also being able to balance that with the clinic. So this is a clinician scientist, or basic science with a clinician side. You'll see with what he said. And he's a reference worldwide now in cholangiocytes, organoids, bioengineering, and he's going to tell you all about it. So Fotis, thank you so much for being here today, and the screen is yours.
Thank you very much, Pedro. I'm very humbled by this amazing introduction. Thank you for your kind words. Many thanks for this amazing invitation. As Pedro said, I am a hepatologist working in Addenbrooke's Hospital, and a scientist working in the Cambridge Stem Cell Institute. Now, as a liver doctor, my research, as you can imagine, focuses on the liver, and more particularly in a part of the liver known as the bile ducts. Of course, I suspect that most of the people attending the seminars know what the bile ducts are, but as a form of brief introduction for any scientists in the group who are not very familiar, the bile ducts form a network of tubes in the liver that you can see here, which drain bile, which is a product of liver metabolism, from the liver all the way to the intestine, where it helps with digestion. And these tubes start as small branches in the parenchyma of the liver, which keep merging together more and more into a common outflow known as the common bile duct. And of course, we've got the gallbladder, which acts as a reservoir for bile. And the single most important cell type in these tubes are the epithelial cells lining their lumen, which are known as cholangiocytes. And these cholangiocytes are quite important. Not only do they form a barrier to toxic bile, but they're also very highly metabolic cells. They can modify the composition of bile. And what is more important for what I'm going to show you today, these cells have a unique potential for regeneration. These are the cells which repair the bile ducts when they are damaged. And also, in some cases and in certain models, they have been shown to act as facultative stem cells, which can regenerate other cell types in the liver as well, such as even hepatocytes when there is damage. And of course, you can imagine what happens in disease. These cells break down and toxic bile overflows into the liver, causing scarring and liver failure. And this can affect either the small intrahepatic ducts or the big bile ducts, such as the common bile duct, for example, in biliary atresia. And these diseases are quite important. They affect children and young adults. They progress very rapidly. In PSC, for example, we lose 30% of our patients within 6 years of diagnosis. Again, in PSC, cancer incidence is very high. We can get 20% of our patients getting cancer at some point in their life. The incidence and prevalence of these diseases is increasing every year. But what's more important is that mortality from these diseases is also increasing year over year in a time where the mortality of most other human diseases is going down because of the advances of modern medicine. And the only treatment for end-stage disease is liver transplantation. As you can imagine, that puts a huge burden on the transplant waiting list. And what's more important, even transplantation is not a cure, because in 20 to 30% of our patients, the disease will recur. And this is, if you think of cholangiocytes only as bile duct cells. If you think of them as facultative stem cells in the liver, then we're moving to a different kettle of fish because, of course, liver disease is one of the biggest killers worldwide. Now, I hope I have convinced you that the diseases are important. However, our progress in the field has been somewhat limited in when I started my research, at least for certain reasons, because of certain challenges. First of all, we know that until recently, we didn't have very good systems to culture these cells in the lab. Lack of appropriate systems meant lack of good platforms to screen and develop new drugs. And in some cases, where these drugs are completely destroyed and obliterated, we don't need drugs because they won't bring them back. What we need is to somehow either repair the damaged drugs by injecting healthy cells in the liver, or if that's not possible, in the case of bigger ducts, for example, what we would need is to generate a bioengineered organ, a bioengineered duct, to surgically replace the one that has been destroyed. And addressing these challenges is what shaped my research that I'm going to talk to you about. I wanted to come up with a system which will allow me to grow cholangiocytes in the lab and use them to model disease. I wanted to use these cells to understand how the bile ducts regenerate. And ultimately, I wanted to apply this knowledge in terms of regenerative medicine, either in the form of cell-based therapy, injecting cells in damaged ducts in the liver to repair them, or in the form of bioengineering, making bioengineered artificial bile ducts. So I'm going to take you through each and every one of these steps. I will very briefly touch on the generation of these cells, since we have done this quite some time ago. Our purpose here, and we decided, was to isolate, and we decided to work on primary cholangiocytes. Why primary? Because we know that these cells already exhibit a very big regeneration potential. And compared to other cell types, we know that they show maturity, and they also show a favorable safety profile. It's much easier to take primary cells back into the patient. Then what we wanted to do is to expand these cells in culture in the lab, validate them, and then test their regenerative capacity. So this is exactly what we did. We came up with methods that allow us now to isolate cholangiocytes from any sample, surgical excision, ERCP samples, or liver biopsies. As you can see here from our flow cytometry, with our protocol, we get a high purity of cholangiocytes. And what we do then is we take these cells and we put them in organoid culture conditions. And you can see here some of our organoids. You can see at the top what they look like under the microscope. This is electron microscopy showing a key feature of cholangiocytes, such as a single cilium per cell. When we do transcriptomic analysis, we see that they are clustered closely to primary cholangiocytes. However, this is very important for what will come next. You can see already from this analysis that there are differences. They're not exactly the same as primary cholangiocytes. And I will come back and touch upon this. And then we've done a lot of functional assays, and I'm just showing you here alkaline phosphatase and gamma GT activity, which are two of the most well-known functions of cholangiocytes, but we've done a lot of other assays. Now that we were happy with the cells in the lab, the next step was to test the regeneration potential. Now, to do that, I decided to start with generating a bioengineered bile duct, not in the least because the lack of the common bile duct in biliary atresia is the leading cause for transplantation in children. So to achieve that, we collaborated, first of all, with the Department of Engineering to generate a tubular scaffold that the cells could populate. So for that, we used collagen and generated densified collagen tubes. They look more like a cone when they come out of our manufacturing process, and then we cut the top bit and we get left with a tube, which we repopulated with human cholangiocytes, which here we've labeled with GFP. And you can see that within 2 weeks, we get a confluent tube. 2 weeks is a bit of time. So what we did is we recharacterized these constructs after they had been fully repopulated and showed that they retain expression of key biliary markers at the protein and gene level, and they retain key cholangiocyte function. Again, I'm showing you here just alkaline phosphatase activity, but we've done many more assays. And the big question, of course, was whether these bile ducts would work in their native niche, which means exposed to bile. For that, we took immune-compromised mice, we excised the mid portion of their common bile duct, and we replaced it with one of our constructs. All this work has been published. And as you can see here, you can see what our reconstruction looked like immediately after transplantation. And you can see here non-absorbable sutures and one of our scaffolds in the mouse. And you can see the proximal and distal duct of the mouse above and below. And this is what our construct looks like 3 months after transplantation. Our cells are labeled with GFP. So as you can see, our cells remain alive. They express biliary markers, keratin 19 in red here. And what's more important is that the collagen scaffold has been completely remodeled and invaded by host cells. These cells are not green, therefore they are mouse fibroblasts. And before COVID struck, we were advancing this technology to the next step, which was to move from small animal to large animal models and pigs. But of course, because of the pandemic, we had to hold some of this work. Now, at that point, I was very happy that we had established a model to grow cholangiocytes in the lab, a platform, I should say, and we had also shown some proof of principle that these cells have a regenerative potential. The next step was to see if I can use now this model to understand a little bit about how the bile ducts regenerate, whilst in terms of regenerative medicine, I've shown some proof of principle for the common bile duct. But of course, we know that most adult diseases affect the small ducts in the liver, the intrahepatic bile duct. I wanted to see whether it is possible to use these cells, inject them directly in the small bile ducts, which are not amenable to surgical repair. There are too many and too little, and see if I could repair these ducts. And on the other hand, as a clinician, we always do our work in animal models. I wanted to see if there's a way to move closer to human. And again, I'm going to take you through these steps, starting from the mechanistic aspects. So in terms of studying biliary regeneration, as you know, to study the regeneration of any epithelium, the first thing is we need to know what we are regenerating to study its cellular composition. This is particularly relevant for the bile ducts because there was a lot of literature at the beginning of this work already suggesting that cholangiocytes in different areas of the biliary tree, you see here the intrahepatic, the common bile duct, and the gallbladder, are different populations. Why is this relevant even? This is very relevant because we know that in disease, a lot of the bile duct disorders affect only a specific population. For example, the intrahepatic duct, which are shown here in red. That means that there are other populations, for example, the gallbladder, which are spared from disease, which gives rise to the question, if we have cholangiocytes spared from disease in a patient, would it be possible to either instruct these cells to directly regenerate the parts of the biliary tree that have been damaged, or another approach would be to say, if this is not possible for anatomical considerations, this is too far from that, would it be possible to take these cells out, grow them as organoids, and then inject them back in the part that has been damaged to repair it in the form of what we call autologous cell-based therapy. But of course, to understand if this is even remotely possible, the next question is to understand, or the relevant question is to understand the relationship between these populations. In other words, are these populations committed to the compartment that they are originating from? In other words, intrahepatic can only regenerate intrahepatic and gallbladder can only make gallbladder. In which case, what I showed you before would never work because as soon as we put the gallbladder in the intrahepatic, it will not engraft. Is it possible that the cells are plastic and they can move freely between different subpopulations? In which case, what I've shown you before, of course, works. Or another very popular theory with a lot of data in mouse models is that there is a common progenitor, an adult stem cell that can give rise to all of these populations. And this is exactly the cell type that we should be going after. This is what we should be trying to isolate and grow. Now, to answer this question, what we did is we took livers, which were not going to be used for transplantation. We excised the different areas of the biliary tree. We took the gallbladder, the big extrahepatic ducts, and parts of the liver. And then we performed, using the 10x platform, single-cell RNA sequencing on these cholangiocytes. And this is what our results look like. And they are in keeping with the literature. You can see here in different colors, different areas of the biliary tree. This is a UMAP plot where each cell is represented by a dot. The closer the cells, the more transcriptionally similar, as you know. So yellow is intrahepatic, green is common bile duct, red is gallbladder. And what you can see here is this confirms the literature. Different compartments of the biliary tree clustered separately because we have different populations of cholangiocytes. That was all good. The next step was to look deeper into each of these populations, looking for subpopulations and more specifically for stem cells. Here, the results were a bit more surprising. Rather than specific subpopulations, what I could see was a gradual transition in the gene expression of these cells in the signature as we move from the intrahepatic to the common bile duct to the gallbladder. And certainly, I couldn't find a specific stem cell population. To look into this a little bit more, I decided to try and understand what the genes that made up this gradient were. So for this, we performed gene set enrichment analysis and gene ontology analysis. And what we found is that almost all of these genes were genes associated with modifying the composition of bile. Now, why is this important? I thought this was very important because bile constitutes the single most important determinant of the cell's microenvironment. So this is one thing to keep in mind. The other thing is that the composition of bile, in terms of concentration of bile acids, pH, and so on and so forth, gradually changes as we move from the intrahepatic, from the common bile duct, to the gallbladder, the same way the identity of the cell changes. That made me think, is it possible that although these populations are indeed of cholangiocytes, are indeed different, there is some degree of plasticity. Their identity is controlled by their microenvironment. And this is why I see a gradient as bile composition changes and not black and white population. Now, to answer this question and test this hypothesis, I came up with a very simple experiment. I decided to take each of these populations, move them away from their niche, and culture them under exactly the same culture conditions as organoids. And this is what my results look like. You can see here, I showed you before the three populations with separate gallbladder, common bile duct, intrahepatic, and these are primary cholangiocytes. And this is what happens if you put them in organoids in this UMAP plot. They all overlap, and the organoids are the different shades of blue. To make it more accessible, I also put it in a heat map with the top 100 differentially expressed genes. And what you can see is that organoids, regardless of their origin, upregulate the same group of genes, which are culture-related genes, and shut down region markers. And this is why I was showing you the differences in the bulk heat map when I was originally characterizing the organoids. So there are differences. Now, of course, that doesn't show that the cells are plastic. This could just be a process of dedifferentiation. My cells became fibroblasts. To prove plasticity, I needed to show that I can now reintroduce a niche factor and get them back to their in vivo identity. For that, I chose gallbladder bile, not in the list, because it's one of the samples which is most easy to access from patients with cholecystectomies, for example. Again, this is what the results look like. We've got now bile-treated organoids in this row here. These are genes which are gallbladder specific, and what you can see is that bile-treated organoids shut down culture-related genes, which were upregulated originally in our organoids before bile treatment, and upregulate gallbladder genes, and this is primary gallbladder as reference, moving back towards their in vivo identity. So that gave me some clue that the cells were plastic and their identity was controlled by bile. My next question was, how does bile control the identity of the cell? For that, I went back to my bile-treated organoids and performed gene ontology analysis, trying to find which are the pathways which bile activates. And not surprisingly, I found that one of the top pathways is the bile acid receptor pathway FXR. So what we did is we took naive organoids which hadn't been treated by bile, activated FXR by treating them with quinodeoxycholic acid, and show that I can get the cells to assume a gallbladder identity as evidenced by the expression of gallbladder markers, FGF19 and SOX17 here, and downregulation of intrahepatic and common bile duct markers. Now, bear that in mind because we'll come back to this plasticity and identity being controlled by bile acids in a few minutes. Now, If I were to summarize what my findings were so far with regards to the cell, I had found that cholangiocytes from different areas of the biliary tree are different populations, but these cells are also plastic. They can assume exactly the same identity when cultured under the same conditions as organoids, and ultimately they are interchangeable because organoids from any region can give rise to any other region if they see the right niche environment. What does that mean in terms of regenerative medicine therapy? That I can take cholangiocytes from an area which has escaped disease, grow them as organoids, and use them to regenerate an area that has been affected by disease. But at this point, bear in mind that this is all in vitro. This is just a theory. What I needed to do is prove my theory in vivo. For that, I went back to my immune-compromised mice, and what I did is I now damaged their intrahepatic bile ducts by using a toxin called methylene dianiline, which was injected directly in the bile ducts. After I induced damage, I took human cholangiocytes from a human gallbladder, and I labeled them red with RFP, injected them into the intrahepatic ducts, trying to answer two questions. Will my gallbladder cholangiocytes be able to become intrahepatic cholangiocytes when they see the right niche and repopulate the ducts? And secondly, of course, is it possible to regenerate intrahepatic ducts with cell-based therapy? This is what our results look like. At the top, you have immuno for a control mouse, which receives the toxin. You can see that we've got degenerate ducts, keratin 19 in green here, and alpha smooth muscle actin with a lot of fibrosis around the portal vein. And this is what happens when we inject the cells in the mice. Our cells, I remind you, are labeled with RFP. You can see here that we've got regeneration of bile ducts. This is a mouse bile duct. You can see only keratin 19 staining, but no red staining. And this is a duct that has been regenerated by our cells, which is green and red. They engraft in the right place, right next to the portal vein and the hepatic artery. And most importantly, they express intrahepatic markers. And I'm showing you here SOX4 in our RFP positive cells. These cells were not expressing this marker originally. Now, this is all one section. And one of the questions that we were asked very rightfully by reviewers when we submitted this work was, how do you know that this is not an organoid which just engrafted there and you cut it in cross section? How do you know that you are regenerating ducts? So we did whole mount imaging. This is a Z stack, and this is the 3D reconstruction of our mice. Green is all bile duct, red is our cells. You see that we've got regeneration of a significant proportion of a mouse biliary tree. So this shows that regenerative therapy with cells really works in the bile ducts. But this is for acute damage. I have to point that out as well. Now, this is all microscopic. We wanted to move to more macroscopic images. For that, we performed MRI and MRCP on our mice. And you can see here that mice that received the toxin have complete obliteration of the bile ducts. Bile ducts are seen in white in this study. You can see the liver is all black here. This is the remnant of a bile duct, remnant of a gallbladder. And this is what happens if we regenerate them. Of course, we do get some damage or some strictures in small bile ducts in the periphery, but we've got huge regeneration compared to the mice before. As a result, all the mice that received our cells survived, whilst the mice that received just the toxin died with cholangiopathy and jaundice. Now, at that point, I was happy that we had gained some insight on the plasticity and regeneration of the biliary epithelium, and also some insight on the fact that we could use these cells, expand them as organoids, and use them for cell-based therapy. But this was all in mouse. The next question is, could we apply this in human? And of course, I was scratching my head about the right context. We wanted to apply this to an acute cholangiopathy. Our mouse model is a toxin-induced model. We did acute damage to these bile ducts. The best way to where there's a big need and also an acute cholangiopathy is in the context of organ transplantation. To be more specific, we know that one of the big challenges in organ transplantation is organ shortage. There's no doubt about that. How do we deal with organ shortage? We're trying to find ways to use more and more marginal organs. How has that been going? Initially, it wasn't going that well, but we managed to improve our selection criteria. And over the years, marginal organs which are carefully selected are performing exceptionally well. But what does that mean? that because we have to select them well, there's a large amount of organs which are not being used still. So now there's a push to try and utilize organs which we couldn't use before. One of the ways to do that, excuse me, is using ex vivo normothermic perfusion. Now, most of you will be familiar with the setup, but again, in case someone is not, what we do is we take a liver and we put it in the bucket of this machine. And what this machine does, which is called an ex vivo perfusion machine or an OrganOx machine, is it circulates blood, which is warmed up to body temperature, going through an oxygenator. So the organ starts seeing warm oxygenated blood as it comes out of the bucket of ice and starts recovering. And what we can do is we can measure organ parameters. If the organ recovers well, we go on and put it in the body. If this organ does not recover, then unfortunately we have to discard it. What we wanted to do, and the most common reason, the most common risk that is associated with one of the organs not recovering is damage to the bile duct, the risk of ischemic cholangiopathy. So what we did is we thought, can we take one step further? Can we now see if some of the organs that do not recover on OrganOx, we could take them, inject them with our cells, just as we did with the mice, and rescue them, showing proof of principle for regenerative medicine in the context of organ transplantation. So this is exactly what we did. I've already told you about, excuse me, the setup of our OrganOx machine, warm circuit, pump, oxygenator. What's more important is we also have a catheter in the bile duct. Again, remember the mouse experiment? We did exactly the same. We took gallbladder cells from a different donor here, labeled them with RFP, and then injected them down the catheter in the bile duct of this liver. Now, injecting a liver with cells means a human liver that you have to have a factor in. We couldn't do that. But what we did have is interventional radiology. So what we did is, as you can see here in this cholangiogram, we went to a terminal, this is our catheter, a terminal branch of the bile duct, inflated the balloon, injected the cells there, kept the balloon inflated until they engrafted, and then we sampled this area. And as you can see here, following injection, our cells, which are labeled with RFP, engraft, regenerate the bile duct. You put keratin 19 here, so you can see that we've got parts of the bile duct which had been damaged and regenerated by our RFP positive gallbladder cells. And these cells lose expression of gallbladder markers, SOX17, and upregulate intrahepatic markers, SOX4, validating our previous results. Now, in human, it was much easier to ask another question. Okay, the cells engrafted, are they functional? In mice, we proved that they are functional because the mice survived. In human, these livers can only be maintained for so long outside the body. So what we looked at was bile parameters. We know that cholangiocytes secrete bicarbonate, and indeed we saw that when we selectively cannulated the right side of the liver where we had injected the cells versus the left where we hadn't, we saw an increase in bile pH due to bicarbonate secretion. We saw an increase in bile volume. Cholangiocytes have aquaporins, they secrete a lot of water, so more choleresis after successful regenerative therapy. And something we established in Addenbrooke's, we have shown that when you have viable cholangiocytes in the bile duct, they eat glucose. So the glucose in the bile is much lower to the glucose in the serum, and this is what we saw, an increase in the difference in blood to bile glucose, showing that these cells were functioning. And now, I've taken all this time talking to you about regenerative medicine. This is where we are with our regenerative medicine program. And as I've said, we're now pushing to generate human-sized bioengineered bile ducts. But at the same time, as I mentioned, I wanted to make sure that we can use these cells, these organoids, as a platform to screen drugs for diseases. And I'm going to show you an example of drug screening in the context of COVID, because to be honest, in the last 2 years, during the pandemic, we had to reshape and refocus all of our research on COVID-related work. And this is what the bile ducts taught us about COVID. So there's little point in talking to you about the challenges in COVID-19. We know that vaccines are limited to prevention, they're not a treatment, and we know that in some cases, such as the new Omicron variant, they are less effective. The treatments, especially at the beginning of this project, showed limited efficacy. And of course, we've got new waves with new mutants, and you saw what happened with the Omicron variant. So there's a pressing need for new strategies, both in terms of prophylaxis, but also in terms of therapeutic. So in order to achieve that, the question is, what do you want to target? There are multiple aspects of the infection that you can target in the viral replication cycle, but we thought that one of the most appealing targets was the viral receptor, ACE2. Why? Because it is needed. Without that, absolutely the virus can't get into the cell. Because we know with monoclonal antibodies that targeting the viral spike protein ACE2 axis is efficient and effective. And of course, because ACE2 is expressed not on the virus, but on the host cells, it doesn't get mutated. Therefore, it's less prone to viral resistance. Now, to do that, both us and others were looking at the best cellular models to study ACE2. And so we did a study with Sarah Takeman, which was published in Nature Medicine, looking at which tissues and cell types express ACE2. And of course, you would expect it to be very highly expressed in the nasal and airway cells. And then one of the other types that seems to express ACE2 at the highest levels, believe it or not, is cholangiocytes, as you can see here. And not all cholangiocytes, and that's quite important because it will play to the plasticity data that I've been showing you before, but specifically gallbladder and large duct cholangiocytes. The intrahepatic seems to express it very low. So what we wanted to do is see if we could now use our organoids, since cholangiocytes express ACE2 at high levels, to identify mechanisms controlling ACE2, see if we can downregulate it and block viral entry this way, exploit this mechanism, as I've already told you, to block viral entry. And then if we found something that was good, if we could then go on and validate its physiological relevance and explore even if we could translate it rapidly to human. Now, as I mentioned before, what we had noticed is that not all cholangiocytes express ACE2 at high level. Specifically, the gallbladder is the area of highest expression. That, combined with what we had found before about the plasticity of cholangiocytes, gave rise to a hypothesis, which was very simple. As I've shown you before, if we increase bile acid concentration, we can get cholangiocytes to assume a gallbladder identity. I've already just told you that gallbladder identity equals high ACE2. We put 2 and 2 together and ask the question, is it possible that if we increase bile acid concentration, we can also increase ACE2? So we did exactly this experiment to begin with. We took cholangiocyte organoids, we checked the expression of ACE2, and we see that in vitro, when they don't see bile, ACE2 is very low in cholangiocyte organoids, and this is consistent with work done by the Clevers lab. And then what we did is we treated them with bile. And lo and behold, we found that as soon as we expose them to bile-lentino deoxycholic acid, the organoids will start upregulating ACE2. This is single cell RNA sequencing analysis, and this is some immuno to see the same at the protein level. Of course, chenodeoxycholic acid is a main ligand for FXR. So we took one step further looking at mechanism and asked the question, is it possible then that FXR is responsible for the upregulation of ACE2? To do that, we performed CHIP analysis showing that indeed FXR binds directly on its responsive element on ACE2. And I should have said before that we found that there is indeed a responsive element for FXR in the promoter of ACE2, and then we performed CHIP to find that it binds and activates ACE2. But of course, we don't want to express more viral receptor and give more COVID to the cells. We need to do exactly the opposite. So we asked, in the context of the pandemic and the need to repurpose drugs, do we have any drugs that can do exactly that, reduce FXR activation? And actually we do. We know that ursodeoxycholic acid, although there's a lot of discussion about the semantics on whether it is an inhibitor or not an inhibitor of FXR, it reduces FXR activity because it displaces chemo. And also, we know that there are other compounds. Z-gugulsteron is a compound which is used by bodybuilders, and you can buy it over the counter, that have the same effect. So we treated ourselves with these compounds and showed that urso or Z-gugulsteron reduce binding of FXR to ACE2. So this is very good at the transcriptional level. Does that mean that we actually get less ACE2 in the cells? So we performed exactly the same experiment and looked at ACE2 expression in our cells by qPCR, protein level, and flow, showing that in cholangiocytes, if you reduce FXR activation, you reduce ACE2 levels. Now, then we started asking ourselves the question, of course, what is the relevance of this? COVID is a lung disease. However, we think that this was very relevant for two reasons. First of all, there are circulating bile acids in the blood. And second, FXR is a receptor which is involved in many cell types, including lung cells. So we decided to go one step further and ask the question, could this be a more universal mechanism starting from the cholangiocyte? So what we did is we grew lung and gut organoids and repeated our experiments, looking at gene expression, flow cytometry, and protein level, ACE2 always in red, showing that FXR inhibition reduces ACE2 level. And this was, again, very encouraging. We were starting to get all excited. It could be a more universal mechanism. But of course, we had shown nothing about the viral infection. So the next natural step was to see if we can now, by reducing ACE2 levels, get reduced viral entry in the cell. For that, we took all three lineages, lung, gut, and gallbladder, treated our organoids with Z-glucerone and ursodeoxycholic acid, reduced ACE2 levels, and by reducing and infected them with SARS-CoV-2. And what we showed is that the reduction in ACE2 level corresponds to reduced infection, as you can see here in qPCR for viral genes. Now, that was all in vitro. The next step is, could we go in vivo? In vivo, we decided to use mice with a caveat that I'm going to explain in a second. So we took mice in collaboration with a Trauner lab in Vienna. We treated mice with ursodeoxycholic acid and looked at ACE2 expression, either at RNA level or protein in the lung, liver, and gut, showing that there is indeed a significant reduction in ACE2 following treatment with ursodeoxycholic acid. However, the caveat in mice is that they are not infected by COVID. So we had two options here. We could either use a Syrian hamster model, but there's only one lab doing that in the UK, and the queue was huge. So with the amount of compounds that were being tested by the pandemic, we're still haven't got the opportunity to get some results from that. Or the other way to test if this is relevant is to see if we could go in a human organ. And for this, we used our ex vivo normothermic perfusion setup, but in this case, adapted to a lung. So what we did is we took a pair of human lungs from a donor, and we split them to right and left. And then each lung was ventilated and connected to a perfusion circuit, which started circulating perfusion solution. Here, there's no oxygenator because, of course, the lungs are oxygenating the blood. So the perfusion solution is just warmed up into body temperature. We performed this experiment, and of course, one lung received carrier control and the other lung received ursodeoxycholic acid. What we show is that we have, after 6 hours already of perfusion, we have a significant reduction in ACE2 levels. The next question was, what does that do to infection? So we infected these lungs with SARS-CoV-2, and what we showed here is that following, perfusion with ursodeoxycholic acid, we get much less viral infection. And now that was all very relevant, and we were very, very pleased with these results. But I'm a hepatologist, and this was an ex vivo organ. So I started asking the question, can this be relevant in the context of liver transplantation as well? And here, we need to ask a very specific question. It is very unlikely that you will get a COVID-positive organ in a COVID-negative patient. I mean, we could ask the question, but it would be an academic question, as they say. What is more likely is that you will get a COVID-positive paracetamol overdose, which will need the liver. And in that case, if you put a healthy liver in a COVID-positive patient, is there anything you can do to reduce infection of the graft by the virus? So for that, we took a liver. We didn't take one, we took actually 3 livers. And we perfused them on our ex vivo normothermic perfusion setting as before. And just as before, we perfused them with ERSO versus not ERSO, just carrier. We infected them with COVID. And what we showed is that even in a COVID-infected circuit, if you put a new healthy organ in, you reduce the chances of you reduce viral entry if you pre-treat with ursodeoxycholic acid. That was, of course, only 12 hours of treatment. Imagine what would happen if we could go all the way longer to 2 or 3 days. And this is all animals, this is all ex vivo. We wanted to take one step further and see if we can translate that to humans. So for that, we collaborated with Ansgar Lochse in Hamburg. And what they did is we managed to get 6 volunteers to take ursodeoxycholic acid. Before they started taking ursodeoxycholic acid, they swabbed their noses. And what we could do is use these swabs to measure ACE2 levels in the nasal epithelium. Then they took, and you can see the starting ACE2 levels, and you can see there's a lot of variation between individuals in ACE2. And then we started giving them ursodeoxycholic acid for 5 days and measured the levels of ACE2 as they were taking ursodeoxycholic acid, showing that indeed there's a reduction in ACE2 in the point of entry of the virus. And then finally, we did a washout period to make sure that this was all ACE2 ursodeoxycholic acid dependent. And what we saw is that as soon as they stopped taking ERSO, ACE levels were restored. Finally, that was something that shows us what happens to ACE2 level, not what happens to viral infection. Well, clearly we couldn't infect patients with a virus, but here again, the fact that we are hepatologists came to our rescue. We decided to capitalize on the fact that all patients with PBC are on ursodeoxycholic acid. And so there will be some patients who got COVID with chronic liver disease on ursodeoxycholic acid. And these patients will be recorded in the Hep COVID or Secure Liver registry, because we do have very good registries, which were developed during COVID. So we accessed these registries and asked the question, how do patients fare in terms of outcomes who have COVID, in terms of COVID outcome, when they are on ERSO versus when they are not on ERSO? And we saw that we get significantly less hospitalization and all the downstream sequelae follow from there. So less ICU admissions, less ventilation, less death. And of course, there are a lot of confounding factors. So these patients were propensity score matched for sex, age, child Q score, severity of liver disease. But we've done further correction since then for diseases that we think contribute specifically for BMI and diseases that we think increase your risk of dying from COVID, such as fatty liver disease. And the results are consistent. We saw that with the caveat of the fact that all patients on ERSO have PBC, still we saw some indication, put it this way, that if you are on ERSO, it could have a prophylactic effect. However, this is not a clinical trial. This does not prove it. It just gives us an inkling and an indication that we should probably proceed to a clinical trial. So to wrap this up, I've already, I think it's become obvious what I've been telling you about FXR. We used our cholangiocytes as a model to find out how FXR directly controls ACE2, test and repurpose new drugs. And there are implications even potentially for organ transplantation from this study, especially in the context of liver, from what I've shown you. And most importantly, we have shown that patients who are taking ursodeoxycholic acid may have a better outcome from COVID-19, which would justify a clinical trial in the future. Whilst if I were to take a step back and look at everything that I've told you so far, I've shown you a little bit on background of the organoid models that we developed. I've shown you how we can use them for disease modeling and drug screening to study mechanisms of biliary regeneration, how we can use them for organ bioengineering and regenerative medicine applications. And also, I think I touched on this new idea of using regenerative medicine, not only directly in humans, but also in combination with transplantation to refurbish humans, organs actually, that will be transplanted in humans. And with that, I want to thank everybody on this slide. This is a massive collaborative effort. I'm just a member of a very, very big team. And I would like to thank you for your attention and take any questions. It's a very good question. So we looked long and hard at that, and actually, we were asked the same thing to interrogate these stem cell markers by the reviewers. Some of the markers, like SOX9, is generally expressed in the intrahepatic tracts, like a lot of cells express it. So I know that SOX9 has been used as a peribiliary gland mark, but in my hands, that wasn't just in the peribiliary glands. You can see it in a lot of the intrahepatic tract. There's a lot of work by Frederick Lemegger showing its importance for biliary development. Some markers, like NANOG, we never saw. Some other markers could be so, and so I have to say that some of the markers that were reported to be specific to stem cells, which were much more broadly expressed in all the cholangiocytes, whilst some others we didn't find, like LGR5, for example, which was not a peribiliary gland mark, of course, but it is one of the other stem cell markers that have been suggested. So we tried to understand this and tie these two findings together. I think it actually fits in the plasticity story. If we think that the identity of cholangiocytes is determined by their niche, it is possible that this stem phenotype is coming on when the niche is becoming more aggressive, if you see what I mean. Mm-hmm. So it is possible that if you have injury, then there are signals that come out that activate this stem program. It is possible that the plasticity of the cells is a continuum. Because we looked at healthy cells, we looked at the beginning. And then as you progress to disease, more and more stimuli come on that open the plasticity so that the cholangiocyte initially can regenerate only itself, then the areas adjacent to it, then the liver parenchyma, and it becomes a bipotent stem cell. And who knows, because there have been some reports that they could even regenerate these peribiliary gland cells, the pancreas. And this could also be true if they see the right signals. But in our livers, which have the caveat of being, well, as you can call them, healthy maybe, we didn't specifically identify these cells. We also have to be very pragmatic and say that single cell is a method which captures cells, but you have to have a minimum number of cells. You have to have a certain abundance. The other possibility is that we didn't catch them. They were there. They were like 10 cells only, and it was below the threshold of our analysis and our resolution. But definitely it wasn't in our organoids either. Yes, that is true. So I don't have an answer which is based on experimental data. We're trying a few approaches, but I think in terms of practicalities, from a medical viewpoint, for me, the most appealing way would be through ERCP. Now, the question becomes, how are you going to get the cells not to be washed out? And this is something that we are trying to develop, because I agree with you completely. It's much easier to refurbish an organ, because the organ is sitting upside down with a hilum up in the bucket, right? So gravity just puts everything. I think that if we infuse the human liver in a bucket, I think that would be achievable. The ERCP is what I want to be able to deliver, but I don't have a very good answer based on data yet. I think it's a question of what we want to try. For example, priming with factors that upregulate integrins or adhesion molecules could be one way. As an example, scraping the local biliary epithelium or washing could be another way. Coating the cells, or even pre-treating the cells in the lab so that I express more adhesion molecules. And I suspect, to be honest, Pedro, that whatever we do, we will not get a percent, you will not get a one go. I injected my cells and now I've got 100% repopulation. I think that this would have to be ongoing infusions. Like we will start, we'll get some in, then the patient will come the next month and the next month and the next month. Or the other option that I would be thinking of is PTC If, however, it is very difficult, however, to do a percutaneous cholangioscopy if you don't have a dilated system. So this is something we are discussing in Addenbrooke's, but I don't know. I mean, tell me what you think as well, because you've got a lot of experience as well. But I think PTC could work, but in a non-dilated system, it would be difficult to get in the duct.
I think the problem is really gonna, if you have to push them through the very intrahepatic bile ducts, the pressure that you need to exert to get the cells there, if you want to get to the whole liver, I think that's going to be a very technical or a significant technical difficulty. So you might have to do it the way you were saying. You have to do it in phases, so injection by injection, and you're going to be regenerating different areas. Because that you can do. You can access the intralobular.
Yes, exactly.
And then those you can treat, segment to segment or lobule to lobule. Maybe that way, because again, you need to have a certain time of incubation.
That's a good idea, actually. That's a good idea. I think you're right, segment to segment. And because you can only get to the second order bile duct, but if you combine it with an interventional radiologist, you could definitely get much further. So I think you're right. It's a good idea. We'll try it. So I think there's a lot of literature on getting hepatocytes or hepatocyte-like cells, at least, from cholangiocytes. I think it goes into the previous question that was asked by you, Pedro, that how much the cell is plastic and how much does it become a stem cell. I think we would have to find the conditions to unlock its stem cell potential. There's a lot of good work done by Mary Hook on this area, and it is very possible that if we use these conditions, we unlock the sort of hepatocyte potential of these cells. To be honest, because we know that this is work which is done heavily by the Clevers lab and the Hook lab, we specifically don't ask that question because it's their field and we want to respect that. But I suspect that it is very, very possible. So I think there's more and more technologies coming out from industry in terms of culturing organoids. I think the first automated organoid culture machinery is coming out. From my point of view, so you can definitely play, we played this in Matrigel and we played this in domes, right? So could this be grown in chemically defined hydrogels? They could. The problem with chemically defined hydrogels is that they're really not compatible with large-scale production because they are PEG-based, and then taking the cells out is a disaster. It's also very difficult to plate the cells because they are much less forgiving. It's all chemical, the cross-linking of the gel, and it happens very, very quickly. You need very skilled technicians. If we were to say that the hydrogel problem will be solved by groups that work on hydrogels, well, I think there are two ways. One would be to use bioreactors. I think Bach has used that, which are flasks, right? And these large-scale bioreactors do work, and the organoids can proliferate quite a lot in them. So we are using a little bit of that, but for us, what really works, the simplest way to do it, to be honest, is that you can get a 15-centimeter dish and just plate so many domes in it. So as a matter of fact, whenever we needed large number of cells, what we found is that the more reliable way to do it was to just plate a lot of domes in a format where we can flood the plate with media, because the plating can be done by a robot, right? And if you've got a large enough surface to create an array, what you can do is you can put media on top. You do one media change effectively, and then it's very easy to harvest them because you can take a very large cell scraper and just take everything out. So for us, it's either big flasks or big dishes that works best. Because I think if you do a big 3D blob, like a block, penetration in the middle for the organoid becomes a big problem. So the only other way is to do it in suspension through a bioreactor, which is constantly spinning, but then you still have to have something to increase the viscosity in there.
Fotis, thank you so much for this impressive talk today, for your time, and see you soon.
It's been a pleasure. Thank you ever so much. Bye-bye.
Bye-bye. See you all.